As you inhale and your lungs expand, lipoproteins called pulmonary surfactants are released from alveolar cells. These surfactants reduce the surface tension of the alveoli, which decrease the pressure difference needed to allow the lungs to inflate. This makes inhalation of the lungs much easier. So basically, what they do is, this is an alveolus, and it's lined with a liquid layer. And these are the surfactants on the liquid layer here. They line the whole liquid surface. And what they do is they reduce the surface tension, which means that the alveoli don't collapse on each other even when the lungs are completely deflated. Leave that there. So this study was aimed to determine the mechanisms behind this release of surfactants, which before recently was poorly understood. In the experiments conducted by Rizard Grigorzik, they found that ATP cell signaling was causing the secretion of surfactants, mucus, and other fluids in the lungs. So ATP cell signaling, I'm sure you've all heard before, it's basically when it's a form of communication from cell to cell. So basically the cell releases ATP, and then outside the cell it can either be converted to ADP, AMP, adenosine, etc., and it can communicate with G protein receptors and ligand-gated ion channels and other extracellular receptors on other cells. And then this can stimulate other cells to release the surfactants, but I'll go on a bit later. More interestingly, the physical stretching of the lung epithelial cells, similar to that during deep breaths or coughs, primarily control the release of this ATP. During the experiments, human type 2 alveolar cells, these ones here, yep, that's these ones, they're the actual ones responsible for secreting the surfactants into the alveoli. So it was these cells that were put into a silicon chamber and stretched by a computer-controlled machine. The ATP release was visualized using a light-emitting substance called luciferin, which undergoes an oxidation reaction in the presence of ATP. And this same reaction is responsible for the characteristic glow in fireflies, for example. So basically, they used luminescence imaging, which you can see here, and live cell imaging to simultaneously monitor stretch amplitude and ATP release. So basically, this is one of the diagrams that was in the results section of the paper, and this is before the stretch, and this is just after the stretch, and then you can see in intervals where the ATP is being released by the orange fluorescence. So it's actually oxidizing and creating that glow in the epithelial cells there. So this ATP is then recognized by purinergic receptors on other type 2 alveolar cells and simulates the release of surfactants on them, and so on. When repeating the experiments with columnar epithelial cells, they found that a stretch amplitude of more than 10%, which is equivalent to that experienced during a deep cough or breath, is enough to cause an ATP release that is efficient enough to signal goblet cells to secrete mucus. Now, goblet cells are just modified epithelial cells specialized for producing mucus in the lungs. They also discovered that the number of cells that release ATP grows with the stretch amplitude. This may be to assist in clearing the lungs from any foreign particles that could be pathogenic or allergens, for example. Another interesting finding was that only a small portion of cells responded to any one stretch, and this cell response was inconsistent and random as such. Now, Grigorzik stated that making sure only a few cells release ATP is probably important, as too large a flood of ATP could overstimulate mucus and fluid production, which could potentially drown the lungs. Thank you.